Hello everyone, welcome to this session. Today we will be dealing our second part of leg operon, and today we will be dealing about the structure of leg operon. See, in the first point it has been written that the leg operon consists of three types of genes which we have discussed in our previous class, that are the leg Z, leg Y and leg A. These genes are transcribed as a single mRNA under the control of one promoter. That means for these three types of genes, one promoter is controlling for the process of mRNA. Now, in the second point, it has been written that gene in the leg operon specify proteins that help bacteria to utilize lactose. That means in the leg operon, the genes which are produced, they are responsible for the breakdown of glucose. Now we will see what are the functions of these three types of genes. These are the genes. Leg Z codes for an enzyme that is beta-galactosidase, and the function of this enzyme is it cleaves the lactose into glucose and galactose, which is the simplest form of lactose. And the leg Y codes for the enzyme galactosidase permease, which is a membrane protein which is required for the active transport of lactose into the cytoplasm of bacteria. And leg A codes for the enzyme that is thiogalactosidase transacetylase, which will help to get rid of the toxic thiogalactosidase. In the exam point of view, it is an important slide. Now we will see the two cases here. What happens in the presence of lactose and in the absence of lactose? In presence of lactose, the first diagram is in presence of a lactose. When this is when the DNA is transcribing the mRNA, which will produce the repressor. Wait. See, we will be dealing with this slide now. Now see, here it is a DNA and it is having a leg operon which will code for the enzyme which are responsible for the formation of the enzymes or the proteins which are responsible for the breakdown of glucose. Here in the presence of a lactose, when the repressor is produced, when the lactose will be present, it will produce a allolactose, for which it will be unable to bind to the operator region. Now the function of this allolactose is to bind to the repressor region and deactivating it for binding to the operator region, for which the leg Z, leg Y and leg A proteins will be produced, which will be responsible for the breakdown of lactose to its simplest form, that is glucose and galactose. Now what happens in absence of a lactose? See, in the second diagram, when the lactose is absent, it does not need any enzyme for its breakdown. So from here translation process is taking place and it is producing the repressor. Here there is no lactose, so it does not have a inducer which is allolactose, which help in the binding of the repressor. So now repressor is free and it binds to the operator region, for which the promoter in the promoter region the RNA polymerase will come and bind, but it will not allow for further transcription to take place as the repressor is binding in the operator region. Now in our next slide we will be dealing with the cap protein and what is negative regulator. See, in the first point it has been written that promoter is a binding site for RNA polymerase, that the enzyme that performs transcription. Yes, the place of RNA polymerase is a promoter, where it will come and bind for further transcription to take place. Now in the second point it has been written that the operator is a negative regulator site bound to the lag repressor protein. That means what happens when the lag repressor is produced, then it will come and bind to the operator region. That is the reason operator is known as negative regulator site, as when the lag repressor will come and bind, it will not allow for further transcription to take place. Now the operator overlaps with the promoter, and when the lag repressor is bound, RNA polymerase cannot bind to the operator and start transcription, which I have discussed now. Now the third point deals that the cap binding site is a positive regulatory site that is bound by the catabolite activator protein cap. When cap is bound to the site, the promoter transcription by helping RNA polymerase to bind to the promoter. That means cap is always present when the concentration of glucose will be low and it will have the lactose concentration high. So for that reason it has to break down the lactose. So the cap binding site is a positive regulatory site that is bound to the catabolite activator protein cap. When cap is bound to the site, it promotes transcription by helping RNA polymerase to bind to the promoter. What happens when cap is bind to the promoter region? Then it will help the RNA polymerase to bound to the promoter region and further transcription will take place. Now we will be dealing with another slide, that is the lac repressor. What exactly is lac repressor? We have these points we have earlier discussed, that is the lac repressor is a protein that represses or transcription of lac operon. That means it is a type of protein which is repressing by targeting to the operator region. By binding to the operator region, it will inhibit the process of transcription. When bound, the lac repressor gets its RNA polymerase way and keep it transcribing from the operator. That means that when the lac repressor is bound with the allolactose, it will allow for the transcription to take place. Now in another point it has been written that when lactose is not available, the lac repressor binds tightly to the operator, preventing transcription by RNA polymerase. If the lactose is not present, it does not need its protein to be synthesized, by which it will be degraded. So that is the reason the lac repressor will go and bind to the operator region, by which further transcription will not take place. However, when lactose is present, the lac repressor loses its ability to bind DNA. That means when lactose is present, and it will produce the allolactose, and the allolactose will come and bind to the repressor region, which will not allow the DNA, which will not allow it to allow the repressor to bind to the DNA region, by which further process will take place. Now the next point is, when lactose is available, some molecules of lactose rearrange themselves in a form called allolactose, which is the inducer. Now in the next slide we will be dealing about some other points, that is cap. What exactly is cap and what is the function of cap? In the first point it has been written that RNA polymerase alone does not bind very well to the lac operator promoter. For that it needs some factors. What is the factor? It is a cap. What happens when the concentration of DNA is low, then the cap protein is activated. So here in this point it has been written that it might take a few transcripts, but it won't do much more unless it's bind to the cap. What happens when the cap is not present? If transcription will take place, it will not synthesize the total transcripts or the total genes which is needed for the expression. But when the cap will bind to the promoter region, then the transcription will be in a proper way, because it will help the RNA polymerase to bound properly to the promoter region. What happens in case of low glucose, when the concentration of glucose will be low? See, here we will see that when glucose level are low, CAMP is produced. The CAMP attaches to cap, allowing it to bind to DNA. Cap helps RNA polymerase bind to the promoter, resulting in high level of transcription. What happens? The cap is the inactive form. It will be activated with the increase of CAMP. Now, where the CAMP will increase? When the concentration of glucose will be low and the concentration of lactose will be high, CAMP will be activated and it will further activate CAP, which will go and bind to the DNA segment, by which the RNA polymerase will help for the further transcription or high level of transcription to take place. Now, we will see the case of high glucose. What happens when the concentration of glucose is high? It has been written that when glucose level are high, no CAMP is made. So, if CAMP is not made, then what happens? The CAP cannot bind to the DNA without CAMP, because it will be in a inactivated form. So, when CAMP does not bind to the DNA, there will be no transcription, or if transcription is taking place, it will take place in a low level, which will be not beneficial for the conversion of lactose into glucose or galactose. Now we will see what is CAMP. It is a sort of hunger signal made by E. coli when glucose level is low. CAMP means, see what happens here, when the CAMP is produced, it is signaling that the glucose level is low, for which it needs for transcription. If glucose level is low, then the lactose concentration is high. For breaking down of lactose, further transcriptions will be taking place. So CAMP is a signal to determine that the glucose level is low. If CAMP will be activated or CAMP will be produced, it will activate the cap. When cap will be activated, it will go and bind to the DNA region. When it will go and bind to the DNA region, it will initiate the binding of the RNA polymerase to the promoter region, which will help in the further transcription process at a high level, for which they will get the proteins which are responsible for the breakdown of lactose into glucose and galactose, by which it will get the simplest form, that is glucose, which will be utilized for the metabolic pathway of bacteria. Now we will be dealing in the next slide in a diagrammatic way. What happens when the CAP is present and when it is absent? In the first diagram, see, in the absence of CAMP, CAP does not bind to the promoter. Transcription occurs at a low rate. See, here is the RNA polymerase, it is binding here, but as CAP is not present in the DNA region, because why it is not present? Because the concentration of CAMP is absent here, for which the cap is in a deactivated form. That is the reason it cannot bind to the promoter region. And the RNA polymerase will be bind to the promoter region, and if transcription is taking place, it will take place in a low rate. Now in the second case, we will see CAP complex is produced here, stimulates RNA polymerase activity and increases RNA synthesis. What happens when the concentration of glucose will be low and the concentration of lactose will be high? CAMP will be activated and it will bind to the CAP, which will help to bind this dimer or this complex to the promoter region, and the RNA polymerase will be transcribed and the activity will be increased. Now in this part C, in the presence of CAMP, CAP binds in the promoter and increases RNA polymerase activity. Here what happened? Here even in the presence of CAMP, CAP complex, RNA synthesis is blocked when repressor is bound to the operator. See, here in this case, when the CAMP and CAP is bound in the promoter region, here further transcription will take place and it will take place in a high rate. But when the repressor molecule will come and bind to the operator region, even in the presence of CAMP and CAP in the promoter region, it will not allow for further transcription to take place for the formation of the proteins. Okay. Now we will see with some other slides. So, when will be the lac operon turn on? In this point C, the lac operon will be expressed as high level in two conditions. When the glucose will be unavailable and lactose must be available. What happens when the glucose is unavailable? CAMP will bind to the cap, making cap bind to DNA, which we have these points which we are discussing now, we have discussed earlier. Bound CAP help RNA polymerase attach to the lac operon promoter, and when lac must be available, the lac repressor will be released from the operator binding to allolactose. This allows RNA to move forward and transcribe the genes. In the previous slide, in the form of a diagram, we have discussed this slide. Okay. In the next session, we will be discussing about the cases, and that will be the last video of this lecture, of this part, that is lac operon. Thank you.